Sciences here at ASU. I'll let him go ahead. Ken.
Thank you very much. It's my pleasure to be actually part of both this forum as well as in the larger conversation. I'm hoping what I have to say today will actually fit in and provide an interesting additional piece to the puzzle of what has been being presented from a slightly different point of view of what we've heard from the earlier talks. I can't anticipate what the later talks will be, but we'll see how that emerges. So I want to start with what's the motivating factor of both this talk and actually the research work that I'm engaged with. So my lab, my group, since its inception, has been about what drives common disease etiology and outcome. And we're very interested, when I say common disease, things like obesity. And what we know is obesity itself has a whole series of origins, as we've just heard very well presented, and a very complex pattern as to how it emerges in the population. But we're of equal interest in my group of not only about obesity, but then the byproducts, the morbid outcomes of things that actually are manifest in the context of people who, say, have such a phenotype. In particular, we're very interested in these disease progressions, all right? So what we're aware of is that when you have a disease like obesity, there are different paths, different outcomes. And to date, it is very, very difficult, I won't say impossible, but from a physician point of view to actually suggest someone who's presenting to you with an obese phenotype, what is their potential of following a particular morbid pathway? So we know cardiovascular disease is an outcome. We know type 2 diabetes can be an outcome. We know various cancers, but we have no simple way of knowing or straightforward way of knowing which of these you will follow, or when you start down one of these pathways, how far you'll progress. So my group, I'm a geneticist, unapologetic geneticist by training and background and a classical geneticist, is to start this search for some of these unifying threads or predictive threads by starting with the role of inherited DNA variation. Now, why do we start there, and does this sound, given the complexity we just heard, as irrational as it might? Because I'm guessing some of you are going, yeah, why are we going to do that? So one reason is that, interestingly, and for a long time it's been known, that these common phenotypes have significant degrees of heritability. So when I mean heritability, it's defined here, it's the fraction of the total trait variance that's attributable to inherited genetic variation. Now these numbers are recognizably soft, depend on the population you're interacting with, the context, the way the study's been done, but it's worth noting that in every one of them, it's double-digit percentages that is attributable to what you, at birth, bring to the table. So part of our interest in studying this, then, is that this, among all the complex factors we just heard, is one of the unifying common threads of an individual throughout that whole life history. You have your inherited variation from the day you start till the day, at least largely, until the day you die. So it's something that we can actually look at as a relative constant across all this other complex environment. Well, I, like many others, actually was embarrassingly responsible for part of this paradigm, felt that we would actually get a big answer to this question with the advent and the application of the products of the Human Genome Project, our ability to have comprehensive genetic maps of incredibly high detail, and that this problem would just melt away. So now we would suddenly be able to explain and have this handle on the underlying genetically inherited variability. Sadly, and it's been shared with this audience by other speakers, so I won't spend too much time on this, this has not worked out the way many of us who were enthusiasts for it had hoped. The individual, as has been said, the individual loci have minuscule effects. They're not observed across independent studies. Replicability of these, even when you have very large studies, is very hard to produce. And more importantly, for this talk, is that fraction of heritability of that number I just showed that's explained is embarrassingly small. So here, and actually are generous estimates, because again, this varies. If you're going to look about the explained percent variance, depends on how big the denominator was. These are generous estimates of how much of the inherited variability in these traits is explainable by these common variations that we're using. So again, optimistically less than 1 in 5%, and quite commonly smaller than that. So as my children would label this, epic fail, okay? So even though we thought we were going to change the world, we have not, okay? Okay? And you can imagine when I do this at NCI, how unpopular that slide is, as the amount of money they're investing in this. All right, why is this the case? Well, we've heard actually, I think, really interesting talks so far, and I look forward to engaging in a further conversation of this. Lots of reasons. Heterogeneity in etiology, maybe due to local adaptation, due to drift. We can talk a little bit about these, but I think at least our group has been interested in measuring and exploring what's the effect of this latter one, not measuring the interactions. These studies, for those of you who are not GWAS cognoscente, are all done under the complete assumption of independence, and in fact, each locus is evaluated independently, okay? So, as this group is aware and has already been pointed out, complex phenotypes are products of networks. Complex interactive networks with feedback loops, other sorts of things that we've heard already discussed. So our group has been interested in using and creating methods, models, and statistical ways that we can use the networks when we do these large-scale genome class analysis. So in using the networks, we integrate regional heterogeneity, so local variation is all rolled up into one network score. We capture explicitly the interactions when we do our statistical models. And for those of you who are stat-oriented, sort of a freebie, but very valuable, is you reduce the dimensionality. So now instead of looking at 1 million independent tests, you look at thousands of independent tests. So you have reduced the dimensionality sometimes by 2 or 3 orders of magnitude, so you get more statistical power. So I will not have real time to go into our methodology of this. I'm happy to discuss this with anyone who's really interested. It's based on a postdoc's work for me when I was at the NCI. The analysis I'm going to be describing here is the pathways of distinction analysis. It's a way that we actually use distance-based metrics to combine and include not only the individual variability, but how the variability is working in concert in a network diagram. And here's a brief discussion, but for time's sake and probably for many people's sanity, I promise I won't go too deep into this. So what we've been doing then is exploring networks in complex traits. And I'm going to go through very quickly 3 sets of interdependent studies that we've been doing. 2 of them leverage publicly available data sets. And the key to this is not just a public data set, but multiple public data sets. And then follow this up with a collaborative data set that we have access to. So I know this is largely unreadable, but it's an obligatory slide from my discipline to describe in this type 2 diabetes what the data sets look like. They have thousands of individuals, cases and controls, a prerequisite for doing a good case control GWAS study. They measure the entire genome. They have hundreds of thousands to millions of SNPs. But you can see we're looking at thousands of pathways and then hundreds of genes within those pathways using this methodology. So in this slide is the output of this, but what's interesting and exciting that I will summarize is out of these thousands of pathways, what we find is 10 that are reproducible across all of the type 2 diabetes data sets. So in this instance, these are all significant. I will point your attention to pathway names that are perhaps as bad as gene names in terms of describing what they do. So we use canonical pathways, sort of what we heard earlier, like the 1 carbon metabolism pathway that we get from KEGG, and we use those as our substrate. But what you'll see, we have an interesting collection of pathways that are signaling pathways, some of these very difficult to understand pathways in cancer, HTLV. One that's particularly surprising to me was axon guidance, and I hope maybe I'll have a minute to be able to come back to that. So axon guidance, let's poke in and see what this one looks like. So here is a similar picture to what we saw for 1-carbon metabolism, just to give you a bird's eye view. If you see green, it was assayed in the SNP chip we looked at. Pink means it was significant in a step down. So we started with looking at all of the genes and variants, and then we did a statistical step down to find the influential ones. And red means that it's common across these 10 pathways. So it's a variant that these pathways are promiscuous. They're not cleanly defined as separate things. So anything that's red is something we see across the pathway. So they may be feeding common modules of common activity. So let's look at one subsection of this. I realize this, just perchance you thought, I thought this was readable. It's not, I realize. So let's blow this one up, okay? So what you can see is one of the modules of axon guidance is the Wnt signaling pathway. And we can see that genes that, again, that were assayed, those that were significant, so PI3 kinase, driving part of the Wnt pathway, and we can see calcium signaling or calcium manipulation as one of the genes that's common across modules. So let's look at another piece of this. So we saw part of this was a Wnt signaling module. Let's look at this one. Interestingly, we see the cytokine or cytoskeleton, which wouldn't probably be surprising for type 2 diabetes, or maybe, and then MAP signaling kinase, or MAPK signaling kinase. So again, similar modules being reproduced. Let's look at a different one, another one that, again, probably you all are immediately saying, what's HTLV1 pathway? So what's in this? Well, let's focus first on this module. Not surprising that we have T-cell receptor signaling, quite interesting, modulating perhaps the immune response. But now in this module, we yet again recover the Wnt signaling and common genes across these networks. Similarly here, we'll focus on here, and we can see another piece of immune response that comes up across these pathways. Okay? So, if we look at all the genes that are common across these pathways, there's 68 of them when we go across these 10 pathways, and then look to see what are their common functions, not surprisingly, given the theme of this workshop and earlier talks, the top 4 categories are all about regulation and control and feedback control systems for these particular processes. So variation in genes that are controlling the controls seem to be critical in determining the onset of this particular phenotype. We also pick up a couple other interesting ones. Immune response, not completely surprising for type 2 diabetes, perhaps, and neurogenesis. And again, I want to put a bookmark for this when we get to conclusions. The other interesting thing, since we said we also can bookend this, we can look at obesity and we can look at liver cancer. So when we look at obesity data sets, we actually see that a number of these pathways are associated with obesity as well. So here I'm going to much more simplify this and not do the combined analysis, but here we see the 3 different BMI data sets we have. We see the relative odds ratios of the contributions of these pathways. One of the things I'll point out to you all is rather than the anemic GWAS 1.1 class finding here, we're seeing that variation in these pathways may actually drive 3-fold difference in the incidence of BMI. And correlated dramatically. So you're seeing big effects. Again, not smoking and lung cancer, big effect at 10, but 3 is actually a pretty significant driver of some degree of phenotype and some difference. All right? Interestingly as well, when we look at the other side of this progression, if we look at the pathways in HCC, hepatocellular carcinoma, what we actually see then is a similar phenomena. So a number of these pathways appear to be progenitor risk modules that may explain why. So again, we can see high odds ratios, relatively small amount of data, but high odds ratios. But what we see then is what may be a potential explanation is why you see certain people in certain tracks. Is that there's an underlying, if we want to go back to control mechanisms, or at least speculated here, is that there's variation or an aggregation of variation, similar to what was said before, that may actually be weakening a particular set of processes that then, when in a different context, puts you either at the edge or the steep edge of failure in moving toward a particular disease phenotype. So let me summarize these observations. The network analysis finds common biologic processes within complex traits. I'll emphasize for a second that when we look at these processes in type 2 diabetes and the others, when we look at the known candidate risk loci, for instance, they are not what are necessarily driving these associations. In fact, the primary data sets that we use did not find the known, not in our hands or in others, did not find those to be the driving factors, even in the data sets. So these biologic modules are related to, but maybe not the same as those basic ones. I think more provocative is that we're seeing these common processes across disease progress, progression paths, and interestingly, they seem to be variation in the controlling processes that seem to be driving a bunch of biologic phenotypes. Some of them expected, type 2 diabetes, obesity, not completely surprised to see a pathway that might be involved with energy metabolism or controlled energy metabolism. Some of them, it may be surprising though, maybe axon guidance or things that are driving neural development, all right? And part of this, again, that I hope we'll have time for discussion is, so why, if there is any evolution driving these, or why would we have these aggregations? And I'm not sure that we have to do anything more than assume this could be, neutral collection of variation that just hasn't been able to be selected against. On the other hand, in the spirit of trade-off, we almost always think of these phenotypes as mismatches, but there's an interesting thread that's been introduced in the evolutionary literature of trade-offs, for instance, with respect to generation of large brains, and the need to actually have both energy metabolism and lipid metabolism be grossly exaggerated in order to physically, developmentally produce the brains we have. And interestingly, things like axon guidance and other of these control phenotypes may have been, or could have been, products of that flavor of positive selection, which may explain why some of these variables are there. They're actually, in aggregation, do permit you to do things bigger, better, faster, but in modern context, mismatch may actually be negative. So with that, I will stop, take questions, and hopefully have intrigued you a little.
Dr. Buetow, very excellent work. I'm Jun Wang from Mayo Clinic. Hi. So one of the challenges that when you consider the pathway, you will have lots of variance, lots of compilation, so computation will be a challenge. I'm wondering how to overcome that.
So there's two answers to that. One of them that I intimated to is by analyzing the pathways, we reduce the computational dimensionality of the problem. So instead of looking at millions of things, we're looking at thousands of things. But I'll answer the question the other way, is in 2017, with computer hardware cheap, you throw processors at it. So we solve these problems by using high performance computing technology.
Okay. So another question is, when you calculate the odds ratio, do you consider any gene is mutated in that pathway? You consider that pathway is mutated, and then versus non-mutated, none of the genes were mutated?
So what we do, and I didn't have time, but I'm happy to spend lots of time. This is like a 2-hour lecture in and of itself, but we actually calculate a score for the pathway, all right, that sums and combines the information of all of the variants in the pathway into a common score. And then based on the score of that pathway, we can see how well that discriminates cases from controls, which is how we get the odds ratio. So it's a combined measurement of variability at multiple loci. Sure.
So Ken, as a virologist, my eyes went to HTLV1.
Yeah.
And with the idea of trade-offs, is it clear that the alleles that lead to risk for diabetes, do they decrease risk for HTLV1, or is there?
So it's never been looked at.
Which direction?
Yeah. So one of the directions I'm interested is actually now getting complementary data sets that would allow you to see what are the risk protection. Can we see directionality, and is there reason to believe that we could literally measure the trade-offs of these?
Good.
But to date, the data sets don't exist.
Yeah.
Two more questions? Jay and then Megan.
I see in one of you had neurogenesis up there.
Yep.
What were you going to say about neurogenesis that you didn't say?
Well, the piece I am intrigued by, and one of the advantages of being for me in the Center of Evolutionary Medicine, is a recent seminar that we had that talked about how these very processes have had to be altered substantially to support the development of big brains. All right. So part of what we're interested in doing is seeing. So we're not initially, when we get things like axon guidance, I can tell you my group gets kind of sick to their stomach and they're saying, oh Christ, so this is obviously not doing anything of interest. So, so part of our interest is to see is whether some of those phenotypes actually might be being driven, might be what are driving the variation patterns. Sort of what Dr. Jakes was suggesting is that can we actually argue that there's maybe positive effects for some of the clusters of these variations for other phenotypes that then when you have them in and why they're common in aggregation is because they're supporting you for something else. But in this context, when you put a bunch of McDonald's on every corner, they actually have a negative output.
Yeah, I just have a question. In your research, have you looked at ethnic differences? Because ethnic differences track these disease outcomes that we're interested in?
So to date, what I would say is most of our studies have been opportunistic. It's the data sets that we can get. So for instance, the data sets that I just looked at tend to be European and American data sets, although fun facts to know and tell is that the liver cancer data set is Koreans, okay? So what we're seeing is pathways predicted as being important in these other European populations appear to also be conferring risks in Koreans. So